good afternoon or morning or evening, depending on where you are. Uh, I'm Shelby Marshall, Vice President of Strategic Partnerships at Fable Vision Studios, and I'm here to introduce Dr. Tomas Helikar and his presentation, Teaching Student Science Through Computational Model Building and Simulation Design and Experimentation. Dr. Helikar is Associate Professor in the Departments of Biochemistry and Computer Science and Engineering at the University of Nebraska. Dr. Helikar's work centers on using computational systems biology and multi-scale models to understand the dynamics of biological processes. He develops software-driven technologies to make computational biology and modeling broadly accessible and integrated with laboratory research and in the classroom. Dr. Helikar also develops new computational biology methods and guidelines as part of several international working groups. Dr. Helikar's discipline-based education research program centers on developing, using, and studying computational modeling and simulations as an immersive educational tool in life sciences courses. Dr. Helikar has built his education research approaches by leveraging his discipline-driven computational systems biology and education software technology development expertise. Dr. Helikar was a keynote speaker at the 2020 Quantitative Systems Pharmacology Conference of the International Society of Pharmacometrics. He's an invited speaker for the 8th edition of the BioNet Visa workshop under the BC2 conference in Basel this coming September. So I welcome Dr. Tomas.
Thank you very much for having me here today. Thank you very much for the great introduction, Shelby. I appreciate it. And thank you to the conference organizers for inviting me to do this presentation today. I also want to add that in addition to being a faculty member at the University of Nebraska, I am a founder of a company called Discovery Collective, which aims at commercializing some of the technology that I will be showing you today to ensure its sustainability and continued growth as it gets more adoption in higher ed or in education as a tech platform, as well as in this biomedical research. And so a little bit of background of what we do. And so we're a relatively large research group at the University of Nebraska. And so my training is in computational biology. And so one of the big pillars of our work is to develop and use computational predictive models to identify more effective and safer drug treatments for different diseases, as well as to understand the complex mechanisms of these diseases. And to do that, we also have a team of software engineers to develop technologies that will enable us that enable us to use and develop these computational models. And then finally, which is the focus of my presentation today, is the use, actually, the same technology that we use for biomedical research. We have expanded on that and have developed it into a full-blown edtech software that gives students the ability to learn about biology the same way that scientists learn about diseases and biological processes. And so I will focus on this pillar of our efforts. And so when we talk about models, predictive models or models in general, we need to realize that models are in fact an abstraction of reality. They're not the reality. And so we all have been dealing with models, whether we realize it or not. We have concept maps that many of us have seen very often, diagrams that can show how, taking a larger process and breaking it down into the individual components and how those components interact with each other, how we get from process A to process B and so forth. We also generate mental models that we may not even think about as models, but we generate ideas and pictures about these ideas in our head, and those are also mental models. If we look at the biological research, scientists use in vitro cell lines to do research to study potential new drug targets, and before they move into, for example, animal models such as mice or other animals. And all of these are different types of models, and they all have their pros and cons, and there are certain assumptions that we make, and there are certain parameters that we need to understand. But the main point is that we need to understand is that they are not the real thing. They're an abstraction of the real thing. And so they have their own limitations, and computational models are exactly the same. They have the same sort of guidelines, and we think about them. We need to be thinking about computational models the same way they can help us with certain things, but they're not the real things. So they have their own limitations. And so today I'll be talking about computational models. And so when I say, again, even in the scope of computational models, there are many types of computational models. You could have a computational model that's a 3D structure of a protein or some kind of object. That is also a computational model. But in this presentation today, I'll be talking about these systems computational models, and they're biological systems that are dynamic. This is a picture of the human metabolism and the different enzymes and reactions that govern that process of metabolism. If we look at other places in our human body, we will find that many of these processes are in fact these complex networks. And so we have developed software called Cell Collective, and it's a web-based application. You can find it at this URL. And we initially developed Cell Collective to help scientists understand these complex biological networks and to use them to identify better drug targets and so forth. And then later on, we have expanded it as a full educational technology. And I'll talk about the sort of the reasons behind that, or how that all came about shortly. And so when we talk about these computational models, so why do scientists use computational and systems models, predictive system models in life and health sciences? And so the answer is that it is because, as I alluded to that just a second ago, it is because biological systems are incredibly complex and they are dynamic. We're looking at a static picture, but in fact, metabolism responds, and our cells responds to cues from the outside, whether it's a medication, whether it's some kind of a pathogen. Our body, our cells respond continuously, monitor and respond to their environment, and they respond by modifying these different interactions and components inside the cells and as an organism as a whole. And so understanding what would happen in a disease state, what would happen if you had one drug in this upper right corner that would target one of these enzymes here, how would that impact something on the other side of this entire system? And this is just one process, metabolism inside one cell. Cells form networks. There are other processes that are equally complicated. The organs in our body form networks. It's all interconnected, and it's networks of networks. And so that is the reason why computational biologists use computational models, or why scientists use computational models. And so the things that we can do with computational models is that we can generate new predictions, and we can also rank our predictions and hypotheses so that when we do go into a animal model study, we are focusing on things and predictions and hypotheses that stand a higher chance of succeeding in the end. And so we can, for example, ask questions. So how will the cell respond if we change the environment? We have, many, many variables. What if we modify those variables? How will the cell react to it? And then we can measure basically anything in a computational model. We can measure any of these components and areas of this complex system. We can ask questions, what would happen if you had a theoretical drug that targets this particular component of the system? How will that impact the rest of these pathways and parts of the network? And running simulation on a computer to answer that particular question takes nearly no time. It costs, once the model is built, it costs very little money to do that. And because it is so efficient and so quick to do that, you can easily start looking at potential combinatorial treatments where you have multiple drugs that you apply to a disease, to a complex disease. You could have one drug that is aimed at that disease and the second drug to potentially suppress the side effects of that first drug. And again, with computer simulations, you can run all of these possible combinations of potential combinatorial therapies in a relatively fast and efficient way. And so if we look at, examples of how systems modeling can accelerate research, and it's important in public health and in society, we don't really have to look too far. We've all unfortunately been through this whole pandemic, and hopefully we're reaching the end of it here. But whether you realize it or not, undoubtedly you have seen many computer models and simulations of computer models behind the hood of these different websites to, for example, simulate different hypotheses, such as how will the pandemic evolve if everybody follows all the lockdowns, restrictions, masks, and so forth? How will it evolve if certain percentage of population does that? How will the pandemic evolve if we limit, certain areas for traveling and so forth? That is all a mathematical model running behind those simulations. And so mathematical models have been used for a long time to answer questions like these, but also at the molecular level, the networks that I was showing you earlier. For example, our group has been part of this very large effort over the last year to, in fact, characterize the COVID-19 disease at the molecular level to better understand what are all the different genes and proteins impacted by the virus, the different cells, again, that communicate with each other, the different organs. And this was part of a very large international effort, collaboration of over 250 scientists from different fields, clinicians, computational biologists, basic scientists from all over the world, and our platform was part of that effort to provide that infrastructure for these computational models to be developed. And as I mentioned before, I won't go over this too much. The complexity in our body that we see is across different parts of the body. Again, genes and cells and the organs, all complex networks. And it turns out that we also continue to learn more and more about different organisms. We keep generating, scientists keep generating more and more data that we have to deal with. And so scientists use computational models to generate new hypotheses, to rank those hypotheses, generate new insights so that they have better insights into the biology and diseases that are then fed back into the technologies and computational models to make them even more accurate. But how do we teach biology? So that's how we've been using, that's how we approach, that's how we learn biology as scientists, as doctors and clinicians. But when we look into the classroom, we don't teach our students the same way. There has been a huge movement to go away from this model where we have didactic lectures and many technologies that have been presented, in this conference too, that help make education more interactive, more engaging, more fun, but also more effective. And in biology, the life sciences field is primed for a change away from didactic lecture and memorization to doing something. And in particular, over the last, I would say, a decade or so, we have also seen universities and decision makers who define standards for our students and what they should be learning realize that in biology, we need to start teaching students modeling and simulation and computational skills. So vision and change is the equivalent to the next generation science standards just for higher education. And vision and change 6 or 7 years ago, maybe as far as 10 years ago, was defined to guide this needed shift away from rote memorization of the individual pieces of a disease or the body, and students need to start thinking about biological processes as dynamic systems. And to do that, they need to be learning about computational modeling, simulations, quantitative reasoning, and so forth. And when we look at the next generation sciences, next generation science standards for a K-12 level, we see the exact same competencies and skills that have been identified as needed so that our students are competitive for today's and tomorrow's jobs. And so, as I mentioned before, so we've developed Cell Collective to be a full research platform and then also education platform so that students can actually learn the same way that scientists learn, and they're familiar with the scientific process right from the beginning. And our idea is that we develop a technology that facilitates the learning of biological systems, not as individual components like we do with these processes. These processes in biochemistry are generally taught as separate in isolation, but they're in fact, again, connected to each other and form this complex network. So it's very difficult to even conceptually separate them because they are all highly interconnected. And so through the technology, our goal is to eliminate the bar to computational modeling and make it attainable to anybody, regardless of their background in computational modeling or computer science programming or mathematics. Just like when we drive a car, we are not expected to be a trained mechanic. We need to understand we need to understand the interfaces in the car, the steering wheel, the pedal, what happens when you press the gas pedal, what happens when you press the brake pedal. But we don't have to be mechanics to be able to drive a car. And so same thing with computational modeling. It needs to be accessible to anybody and without having to have that training in programming. And then with that, through the computational modeling, we want to give students the opportunity to learn and practice through a constructivist and inquiry-based and scientifically authentic way. And so here are a couple of recordings of the Cell Collective user interface. And so as you will see here in the left panel, there is a network being built or drawn, basically. And that's how students build these models. You don't have to have students enter any differential equations or complex mathematical equations. They can simply draw the network like they would do with the concept map, for example, on a piece of paper. And the software is building the mathematical relationships in the background so that when you add these components to the model, they are actually already simulatable. It's not just a static picture, it's a fully simulatable model. And so in this case, we're building here the biochemical engine behind that govern how bacteria move. They either swim forward or they spin around. Those are the two types of movements that they can do, and that is governed by this biochemical network that has some interesting properties biologically, but I won't be going into that. And so when the model is built, then students can, or users, scientists, students can simulate it and evolve that model in time. And so, so in this middle panel here, the activity network, you can see the entire network, and it's changing colors, and it's changing colors based on the different activity levels of the individual components. On the right-hand side of the simulation graph, you see a time series graph, the dynamics of the two selected nodes. So the running motion of the bacterium or the tumbling, which is the random spinning around. You can see how those patterns of those movements change, and they change in response to the different environmental conditions that the student can change simply by changing these sliders, or they can check the little pill box here to simulate a drug treatment and see how that, if you have a drug that inhibits this particular protein, how will that impact the dynamics of the system? And so you can do a lot of what if scenarios. You can play with the system. You can poke and prod it in this virtual way. And so we've developed a Cell Collective now we have a little around 30 different modeling lessons, and we designed the lessons such that no prerequisite knowledge is required, so that we can sort of level the playing field for students, because they may be coming from different backgrounds with different prior knowledge. And so our modules, our modeling lessons are very self-contained, and they provide enough information and knowledge to the students to be able to successfully complete these lessons. Every lesson we aim to be around 90 minutes, but of course that'll vary a little bit. With as many lessons as possible, what we want to do is we want to give students the ability to build their own models. We don't want to give them a pre-built sort of black box where they will change some parameters. We want them to be able to build that model, because by building something, we are more likely to learn how it works than being given that object and then trying to figure out how it works. Okay. It is a web-based application, and so it's been. We also keep in mind the flexibility of its implementation, and because it's sort of modular, it's self-contained. You don't have to change. We don't expect instructors to have to even change their syllabus. They can simply take these lessons and plug them into their syllabi wherever it fits with with the topic that they're teaching. And it being a web-based technology, it can be used either on campus or remotely. We've seen as many EdTech solutions have probably seen a huge growth over the last year, thanks to COVID, and so it can be used remotely as well. We've had instructors use it as part of the lecture, where students complete or begin completing these modeling lessons during the lecture, or they do it as a laboratory assignment in class or as homework as well. And some of the examples of the topics that we have developed these modeling and simulation lessons for are listed here, but you're also welcome to check out the website and the dashboard to see what different topics are available. Our initial funding, so this whole technology has been funded by the National Science Foundation through federal grants to the university and to our research program. And we've been primarily focused on developing these modeling and simulation lessons for initially introductory biology courses. There's usually a 2-course sequence the first year that students enter college. We've developed a lot of lessons for those, but then over time and with additional funding, we've been able to expand the number of lessons and the types of lessons to also cover more advanced undergraduate level life sciences topics and in immunology, for example, advanced biochemistry, cancer biology. But also over the last, I would say, 4 years, we've been getting a lot of interest from high school teachers who have been adopting a lot of the intro biology course modules and modeling and simulation activities in their biology classes. And so we are gearing up to start developing these modeling and simulation lessons that are even more NGSS aligned and age appropriate for younger audiences. And we have one teacher overseas who has adapted, who has adopted one of these lessons or a couple of the lessons for her third graders. And so there's a huge opportunity to really make something like this more accessible even to younger groups. And so here is a overview of how we design these modeling and simulation lessons. So as I mentioned before, a key, very important component and sort of design principle is that students need to be building the system. Whether it is whether they build something completely from scratch with just starting with a blank canvas, or whether it is to connecting different subsystems into a much larger system or expanding on an existing system, then we have students make predictions so that they can start learning about generating hypotheses. We have them justify those predictions to really make them think about mechanistically why they're making the prediction that they're making. And then they use the model to test their prediction. So they have to be able to do that, they have to think about the experimental design with that system. Then they get to report on the simulation results so that we can make them think about data interpretation, and we have them evaluate their prediction. So again, data interpretation, and we want them to go back, and once they ran their experiment, look back at their initial prediction and understand whether their prediction was correct or incorrect. And if it's correct, then great, explain why it is correct. But if it's not correct, now start thinking about why it is incorrect. And that is also this iterative process now that it allows them to think about the system as a complex system, and it enables them to start thinking about the individual components of the system and how they're all connected. And they can go back and revise their predictions, and they can revise their model, for example. Perhaps the model was incorrect. And so they can go in this iterative process until they are sort of satisfied, until their predictions match the output of the simulations and of the models, until they're happy with the system that they build. And so this is an example of one of those lessons where we teach them about glycolysis. And so students are building the model here. Then they have the simulation control panels. They can change the parameters of the model in real time, and then in real time interactively see how the model responds. And also, and I will show you a live demo in just a few moments here. And then we also have students on the same screen answer those questions and kind of take notes. And so we can do also formative assessment as we go through, and also for students to be thinking, making them to think about the system and how it behaves. here's an example of a study. We've published many research articles that show the efficacy and learning gains when using technology like cell collective in the classroom. This study was done very recently, and the paper was published on this in February of 2021. And so in this particular case, we had a setup where we had a upper biochemistry, so senior and junior level biochemistry class at a four-year university. And we were teaching students about glycolysis, the three different biochemical systems, the glycolysis, the TCA cycle, and electron transport chain. I don't know the background of the attendees here, but the biology or biochemistry here is not important. What is important is that we implemented these modeling models and simulations in a couple of sections of the large format biochemistry class. And then we had other sections of the same course, the same year, that did not have this treatment. They did not use these models and simulations. And we had an assessment associated with it that enabled us to measure how much students understand these concepts that we're now teaching through models and simulations. And many of them were multiple true false questions. And so when we look at the results, we did a pre and post assessment. So we asked the same test questions before at the beginning of the semester, and then also at the end of the semester. And you can see the statistically significant learning gains when students used the models and simulations, whereas in our control, where they did not, there was no change in their understanding of these biochemical concepts. And I'm not showing these data here either, or I'm not showing these data here on this presentation, but if you're interested in that paper, we also found that repeated exposure to these computational models and simulations actually cements in the learning gains for students. They achieve more and more, which makes sense because students are learning with a new approach, with the technology they haven't seen. They are not being fed this information through a lecture or having to memorize it through a textbook. And so the more used they are to this technology and to this approach, the more they can get out of it. But it was nice to be able to measure that and capture that. And so today, Cell Collective has been used by nearly 150 different institutions, mostly post-secondary community colleges and four-year universities here in the US, but also overseas. And so we look forward to growing the technology and developing more activities and helping more instructors to figure out how to use these in their courses. So I have about 20 minutes. So what I will do is that I will leave this and I will jump over to Cell Collective so that I can show you kind of in real time how the technology works and so that you can get a better idea than from static slides. Okay, so when you go to cellcollective.org, you can see access through three different sort of points. As a scientist, which we will not be going into, but that's where biomedical scientists go to use this platform for their biomedical research. You can access it as a student, or you can access it, request instructor account, which will give you the answer keys and also additional instructor supporting materials and so forth. And so what I'll start with. I'll go to the student view so that you can get an idea of what the technology looks like from a student perspective. Okay, I'm logged in. So this is the dashboard where you can see the different topics of computational models that we different topics we teach with the computational models. And I'll go to this training module, which is a module that we have instructors deployed to their students before they actually use any of these other activities to teach the actual biology. Because with this technology, there are a couple of hurdles that we've had to overcome. Number one is that it's a new software. It's very different than from what students are used to. And so we need to get them comfortable with the software before we get them to focus on the biological system, on the learning of the biological system and its dynamics. And the second challenge is that the chances are that our students have never used a computational model or a simulation unless they are in a computational biology or bioinformatics major or mathematics major. If they have some kind of an engineering side major or minor, then pure life sciences biology students will likely not have been exposed to computational models. And so we need to get them to frame their mind around what we mean by computational model. As I discussed before, there are different types of computational models. So we need to put their mindset in these models, in these network models. And we need them to understand what simulations are, what kind of information you can get out of these simulations and so forth. And so this this module takes about 30 minutes to complete. Like I said, we have instructors to use it at the beginning, before they start using Cell Collective for any of the biological topics. And so students start the lesson, and then there are different tabs and pages that students are sort of navigated through. And usually the way we have designed these lessons on the I'm going to change the sizes here of these panels, so that on the left side, we give students the scaffolding, the instructions on what to do, but also how to interact with the software. And so this training module, as I mentioned, is to get them familiar with modeling and the technology. And so we specifically built this activity around a topic that they will know something about, right? And this topic is about how to get your exam score higher. What are the factors that can impact your exam score? And so we have students model exactly that. And so we ask them, okay, we're going to build a model of that, and you're going to simulate the model. And so students start building the model by double clicking in here and add in this node, and we'll call it exam score. Okay. And then we ask the students in the instructions to also add parameters, the variables that can change your exam score. And so we talk about studying. The level of studying that you do will certainly impact your exam score. And we also have a parameter called stress, because we know stress can impact what we do. You can move these nodes. You can hold Control key and move these nodes around as you want, position them. And then we ask students to connect them. So, so how does studying impact your exam? And we know that would be positive, hopefully, right? And so we draw an edge between the two. And so the green arrow here says that there is a positive causal relationship between studying and an exam score. And then with stress, we know that it's a negative relationship. So we once again just draw the arrow between the two nodes, and it defaults to positive. And so we just push shift and click on it, and it turns it into a negative relationship. And so now we have two variables here, and two independent variables, and we ask students to think about the independent variables versus dependent variables. And to make these actually independent variables, we have to change their color again, and so we make them orange. And so now we're telling the computer these are independent variables, and we'll be able to control them. For students, we make them think about independent and dependent variables. And then, so students build this model through these few pages, and then we get to a simulation panel. And so in this simulation panel, so we have I will focus on a couple of these panels here. So again, on the left side, we ask the students to, and maybe I'll zoom in just in case the screen is not visible as much. So on the left side, we give students the instructions on how to set up the simulations. And then we have multiple panels here. We have a panel here for the internal components where students can check the observable variables that they want to observe. So we want to observe the exam score during the simulation. And then this is the simulation control panel where you can start and pause the simulation, change the speed and some other parameters. These are our external components. Those are the independent variables that you can change their activity here by sliding the slider. Then we have this activity network where you can observe, once we start the simulation, the network change colors based on the activity level. And then this is our simulation graph where we'll see the exam score or any other variables that you're observing evolve over time. And so we start the simulation and our exam score is at the lowest because we don't have any stress, but we're also not studying. So students get to play with, okay, make a hypothesis. What happens when you study? How will that impact? Your exam score? And so you can see the response of the exam score goes up. It's stochastic, so that's why you see it's not a straight line. Okay, and then we can start asking questions. Okay, well, what happens if you are studying someone but you're a little stressed? Okay, so your exam score may get impacted a little bit, but it's not going to completely, you're not gonna maybe fail the class or fail the exam. But what happens if say your dog died yesterday in a real way, not an excuse, but your dog died and you're just it was your first dog, you absolutely loved him, and he's gone now, so your stress level is going to go through the roof. So we'll turn that up to 100%. And you can see that even if you studied, you are so distracted, you're stressed out that maybe you will fail that exam. And so students get to play with the simulations like that, and then they go through this further on, and we get to here where we actually have, I'm going to keep going here. I'm kind of narrating this module as we go, so I'm not following the instructions here to the T. So I'm jumping around or jumping through some of these pages. Here we actually ask students to add another variable. So you've got studying and you have stress, but with studying also goes note taking. So we ask the students to add another variable, and we'll call that note quality. Okay. And then we have the students think about how note quality impacts the exam score. And so that's in a positive way, right? The better the notes you have, the better the exam score, perhaps, right? But is that it? And so now we ask students to think about the two relationships, for example, between studying and the exam score and note quality and the exam score. What happens if you study a lot, you studied all night, but your notes were not so good? You maybe didn't have any notes. So do you think that would be sufficient to positively impact your exam score or not? And so we walk through this and we come to the conclusion that in order for studying to have a positive impact, we also need a high note quality. And so we have students here start thinking about these mechanisms in a conditional way. So if you have studying, but you don't have good note quality, your exam score is likely not going to be positively impacted. So students do that here, and then they go back and they start and they do more simulations. And so I'll run a simulation. Now we have 3 independent variables here. We have note quality, stress, and studying. Okay, so I'll put all of those to 0, and then we'll start the simulation. Exam score is 0, but now we can see, okay, what if I have studied a lot? It doesn't really help me because I don't have any notes. So let's add some good notes to the mix, and suddenly our exam will go up. And you can ask a question, okay, well, you have good notes, but you didn't study. What happens? And your exam score will go down, right? And so again, students get to play with this. You add stress, and again, that changes variables. So I'm running out of time, but I am also at the end of this demonstration. And so I want to say is that, so this is sort of the approach that we then take on all of these biological topics, so that students can learn biology by building, breaking, and playing with these computational models. And so with that, I will switch back to my concluding slide. And I will be happy to take any questions. Thank you so much.
Oh, thank you, Tom. That was wonderful. That is incredibly impressive.
Thank you.
We'll see if we have any questions, and while we're waiting for questions, I can see I love the way you get students to learn how to use the software and how to think about the software through modeling this everyday behavior. So before they get into understanding what's going on inside of cells, they just get to understand how modeling works.
Mm-hmm.
And it's not just how the software works, but how the whole concept of modeling works.
Exactly.
And I wonder, do you get, I mean, this probably would take a few steps in the experiments, but do you have a point where students can actually define their own questions and design an experiment that will, so here's my question, here's a hypothesis, yes. How do we design an experiment for this and build a model to test it?
Yes, so a full-blown inquiry approach, and it is completely possible. We have some lessons that have that in the end, where students built the model, they did simulations, so they understand the system, and now they are supposed to ask additional questions that cannot be answered with the model that they have. So they have to expand on the model and then start asking those questions and answering those questions. So we have those aspects in some of the activities. We've once tried where we basically gave students, here's a biological problem, a system, and design your questions around it and build a model and produce answers. And it was a disaster, in fact. What we've learned is that it was too much unknown, too many things to think about. It was very different. Students were more frustrated than I think they learned, but they still learned. But I think the frustration component also needs to be taken into consideration, whether students want to take a chance on these innovative learning approaches, because especially in our biochemistry department, we get a lot of students whose number one priority is to go to medical school, and so to go to medical school they have to have straight A's and everything, and so they if they don't see how this learning method will help them get into medical school, they're not interested. And so so we're sort of trying to, with the way we set up these things, we found that sort of that balance of scaffolding, giving students enough instructions but not too much, because we don't also want to make it a cookie cutter recipe like experience. There has to be a inquiry in it, but it has to be, we found that it has to be a good balance of inquiry, independent inquiry, and scaffolding to make sure students are still progressing.
That makes a lot of sense. Do you find that that you need to train instructors in not only how to use the simulation, but how to teach with it?
Surprisingly, not as much as we thought that we would have to. A lot of the adoption of the software has been completely organic. Again, we're at the university. We don't have, our grants will not fund a salesperson, and so we develop this technology, and then we go to conferences like these to share what we've done and how it works. We publish scientific literature on that, and then instructors find those or meet us at conferences, and then they start using it. We've recently had groups of instructors who are interested in learning also not just how to use it, but also how to modify all these lessons, because we've designed Cell Collective in such a way that all of that content that you just saw, it's completely changeable. You can, instructor can go in, make a copy of this lesson and make changes to it, and deploy to their students that changed version, or they can deploy a lesson completely from scratch. And so we've now had more instructors interested in learning how to build their own lessons, as you know, and very little about how to use the existing lessons.
That's really a great result. I mean, it shows that not only is it really useful, but it's easy enough to use, and they're finding enough value in it that it's worth their time to adapt it to their own coursework. It's interesting. The NGSS, the science standards at K-12 and higher education are so similar, of course. But one thing that I'm struck by is how well this teaches or gets students to really into data literacy and computational thinking, which are things that are certainly built into those standards, but tend to be very difficult to teach. To construct an learning experience that really gets students to understand data and statistics, while not being focused on that per se, but having that be something that you're doing in the course of learning something else. It's hard. And here, this is just a naturally part of it.
Yeah, it is hard. And also we see maybe, I would say maybe there has been a shift in students maybe starting to understand that they need to learn a little bit of the technical side of the world, programming and math and statistics and data analysis. But I would still say a lot of students who we meet in life sciences, in biology, biochemistry, they still stay away from a computer science class or a statistics class. They'll take it if they have to, but they still try to avoid it, and they'll still answer. They'll still tell me, I'm not good with computers. This is not for me. But we'd be doing the students a disservice if we did not find a way to teach them these skills, because they are absolutely critical for the jobs, whether they will be healthcare professionals, doctors, or whether they will be biomedical scientists. That these tools are part of it and part of our jobs, and they will become even more intimately integrated into these jobs and absolutely necessary.
Yeah, it's interesting. I was a biology major a very long time ago in the early 70s and took a course in population dynamics. And I was an ecology major. And back then, we were using, Fortran 4 and BASIC. And so population dynamics is, the modeling you could do is fairly simple. And I look at this and I think, well, you could apply these simulations, this modeling to population dynamics, to sociometrics, to all sorts of things that are at the macro level. You don't need to stay inside the cell, inside a body. I mean, it's a marvelous tool for studying any kind of system.
We actually have a food web dynamics modeling lesson and simulation lesson there.
Oh, excellent. Yeah. Huh. Have you tried anything? I mean, this, I don't know if this would be practical, but I look at this as something that could actually be a powerful tool even for citizen science, for things like that folded game, where people are experimenting with protein. Somehow this could be deployed on a broad level to have people explore questions that are
absolutely. One thing that yes. So in the biomedical research field, a lot of talk, and we've been working with a lot of different scientists about crowdsourcing the construction of these models so that we can model more and more accurately and at a deeper level of detail. And the citizen science is an extension version of that. The one thing that we haven't sat down or talked to people who know that aspect, which is gamifying this. For citizen science, I think there has to be a game aspect like Folded has. And I don't know how to do that yet. But I would love to figure it out. And I think that would be one of the missing pieces to turn it into that.
Oh, I'd love to talk to you about that. We've done work with Concord Consortium on game development with that kind of, that would be very cool.
Yeah.
Well, so I think we're getting close to our time. It's 4:28. And we don't have any new questions. Fortunately, this will be recorded. This is so incredible. How long did it take you to develop this stuff?
We started in 2010 when it started as a technology to make modeling accessible to wet lab biochemists who also don't necessarily have computational background, and so we wanted to communicate the models to them. And then in 2014, we started adding and expanding it also into as an full-fledged educational technology. And so over the last 6 or 7 years, that's been evolving, and we've been developing new topics, models, and simulations for new topics.
Wow, wonderful. Well, it shows. Thank you so much for bringing your work to the conference.
Yeah.
And I'm sure that there'll be a lot of people interested in watching the recording and learning more about what you've been doing.
Thank you, Shelby. I appreciate it. Thank you, everyone.